this is the 2025 O-Level TYS Paper 1 MCQ for O-Level Pure Biology. And one of our TBT students sent me his answers. So we're gonna mark and I'm gonna explain each question and we'll see what marks he gets at the end. We're also going to post the answers in our Telegram channel and on our social medias. So in the description of this YouTube video, you'll be able to see the links to the Telegram channel and also just search that BioTutor. You'll be able to follow us on social media. Question 1, the answer is A, release of most energy from glucose. Now, what ways can we release energy from glucose? Actually, there's two. Aerobic respiration, which happens in the mitochondria. That's why A is the answer. And this gives us large amounts of energy. The other way is anaerobic respiration, which releases small amounts of energy from glucose as well, because it also breaks down glucose. Where does it happen? This is a bit FYI, but it's super useful to know as well. It happens here. This is where anaerobic respiration happens in the cytoplasm. So question 1 is A. Question 2, so features of red blood cell and root hair cell. If I'm not wrong, this might be a copy-paste exactly of a past TYS question, which is why we encourage all students to do all the past 10 years of TYS. And in fact, if you want to do more than past 10 years, as a TBT student, you have access to our soft copy of even beyond past 10 years. So because they reuse it like this, you just get free marks if you have actually done the past TYS. Now, contains chlorophyll, red blood cell, obviously no. Root hair cell, no. Even though it's a plant cell, but it's underground, it doesn't care about photosynthesis. Then, contains cytoplasm, yes. Both of them have cytoplasm. It's a very basic essential for any living cell. Now, xylem vessels, when they're mature, are dead, so they don't have cytoplasm, but they are one of the rare exceptions in your syllabus. Now, option C, large SA to V. Red blood cell has this biconcave shape, and this increases its SA to V, so yes. And then root hair cell has this elongated protrusion, and that is what gives it the high SA to V. So that's why C is correct. Okay, then mitochondria is present only in root hair cells. Why? Because they need it for aerobic respiration to release large amounts of energy for active transport. Why? Active transport mineral salts. This is how they even absorb mineral salts. Because inside the cell is high concentration, so they have to use active transport to go against the gradient. Okay, that's how I know root hair cell is yes. Red blood cell is no. Yeah. So why is it no? Because red blood cells care about just packing as much hemoglobin as possible to transport oxygen. So that's why they sacrifice the nucleus, they sacrifice the mitochondria. Question 3, which is true about the process of diffusion? Molecules are always moving is yes, right? So you might have learned this in chemistry that on a molecular level, if you can zoom in the microscope, right, molecules are constantly moving and banging into each other. So molecules are always moving. This is true. Now, the second column, they only move when in solution. And that's not true because, like this student wrote, it can also be happening in a gas. Diffusion happens with gases as well. Think about water vapor, which is a gas, diffusing out of intercellular air spaces. So that is down to concentration gradient diffusion of a gas. That's one example in your syllabus. So this is false. And then the last one, molecules move down the gradient, yes. Okay, so the answer is indeed C. Question 4, which shows the smaller basic unit that forms the large molecule which is at the top in the header. So DNA is made of many nucleotides, so it's either C or D already. Then lipids, aka fats, right? So fats are made of 1 glycerol and 3 fatty acids. Now technically lipids is a big group of molecules that includes fats. So the one that you guys learn is the fat molecule, the one with 3 fatty acids. So yeah, it's fatty acids and glycerol. Okay, that already narrows down the answer to D, so D is correct. Let's just continue seeing the last two columns. So proteins are made of amino acids, yes. And then starch is made of many units of glucose. So that is correct, is D. Alright, question 5. So we see an enzyme temperature graph. We see at X, which is the optimum temperature. And we notice that beyond the optimum temperature, as we expect, the rate goes down to 0 because the enzyme is denaturing. So what does that mean? At X, right? Let's look at optimum at X. So this is the enzyme, okay? And let's say this is the substrate, right? So it fits perfectly into the active site. This is called complementary, right? It is complementary to the shape of the active site. That's why it can fit, and that's why it now forms an enzyme substrate complex. Okay, so enzyme substrate complexes will only form if it can bind, if it is not yet denatured. Now, if we go to temperatures higher than X, some of the enzymes will start denaturing. And if you go all the way to here, then all the enzymes have been denatured. That's why no reaction is possible. So let's say the enzyme has become this shape, right? It is, okay, no, let me do it even worse. Yeah, so it's completely been destroyed. That active shape is like so dead. So this poor substrate, even if it's unchanged, it cannot form any enzyme substrate complex. So no ES complexes will form like that. Okay, so statement 1 says that if you increase the temperature, then the ES complexes form will decrease. Yeah, that is true. Because as you can see in my diagram there, when you mess up the enzyme like that, denatured, it cannot form anymore. It cannot bind, it cannot form the complex. So 1 is true. Now 2 says the shape of the active site and substrate all mirror each other. So firstly, this is the wrong word to use. We should be saying they are complementary to each other. Second, this only happens if the enzyme is fine. But if you go higher than X and you denature, then it won't be even complementary. So 2 is completely wrong. Then 3 says the substrate is being denatured. Now this one is question mark. You don't know. In fact, starch is amylase catalyzes, right? This is enzyme amylase. So if starch you heat it up higher than here, and we know that 37 degrees is the optimum for human amylase, then this point here seems like maybe 50 degrees, right? I don't know if it's to scale, but it seems like it's not crazy high, like 1000 degrees. So 50 degrees will starch denature. We actually don't know, it might be fine. So the substrate, also another thing is denature is used for proteins, right? So starch being a carbohydrate made of many glucose, does it denature? I don't think we can use that word. So I do not think 3 is correct. So that's why I'll choose B. Okay, so the answer is 1 only, B. Question 6, it asks us, why is it that this enzyme only will detect glucose in the blood? Why will it not detect some other random molecules like, I don't know, amino acids, fats? So the answer would be the specificity, right? That enzymes active site are specific to one shape only, which is to the shape of glucose. A is the best answer because it's actually that same concept of being specific. Now, enzymes are not changed by reaction, but so what? You know, it doesn't explain why it would be specific. And then have an active site. Again, what about the active site special? You have to say they're specific to the substrate. Lower the activation energy actually just tells us why you don't need to add extra energy or heat, and it's able to still catalyze the reaction. So it's explaining the wrong thing. Yeah, that's why the answer is A. Alright, question 7. So how does bile work? We need to understand in the first place. Now, fats, when we eat fats, they like to clump together. Because they don't like water, so fat likes to come together into this huge fat droplet, right? Big fat droplets. Now the problem with these big droplets is that larger an object is, the lower the surface area to volume ratio will be. So if I am an enzyme lipase and I'm trying to get a hold of some of the fat molecules, it's very difficult and it's going to be very slow. So what bile does is the bile salts emulsify these large fat droplets, which is actually a form of physical digestion. So what it does up into smaller fat droplets. Okay, just imagine a group of students. They like to bunch together in the class and they like to talk to each other. And then the teacher comes in and it's like, hey, y'all are always clumping together as one big group. I'm going to split you all up. So then it starts moving the students all throughout the class. Okay, so maybe they'll be less noisy. And that is what the bile salts do. So now it's small fat droplets, right? The surface area to volume ratio is much higher, and this gives a lot of opportunities for the enzymes to find a fat molecule substrate and to break it down. So when you break down a fat molecule, you have glycerol, 3 fatty acids, right? Now you get the components, and these fatty acids, like the name suggests, are acidic, so they reduce the pH. Okay, a sidetrack, amino acids are not acidic. It is the only acid you learn in the syllabus, right, that is not acidic. All other like carbonic acid, fatty acid, all these nucleic acid, right, like DNA is acidic as well. So all these things are acidic, they lower the pH, but amino acids, okay, this is just an FYI note, amino acids are not acidic, they're actually neutral. Yeah, so this is the only like scam acid that you learn in syllabus. So now we look at option 1, it says bile increases the speed at which lipase acts on the fat. Now, we can see this from the graph that is true because the pH is decreasing faster, which signals that the fatty acids are being produced faster. So 2 is true because it does decrease the pH. And then 1 is also true from the graph, you can see that. And they all reach the same final pH with or without bile. It's just that one seems like it's reaching it faster. So why do they reach the same final pH? Because the same amount of fatty acids were produced. Example, 10 units was produced in total from breaking down all that fat. So the final amount of fatty acids is the same. It's just, do you produce them faster or slower? But the total amount is going to be the same. So 3 is wrong. So the answer is indeed B. Okay, question 8. There are the biomolecules added into all the test tubes: starch, protein, and fats. And there are some mystery enzymes that we don't know about, digestive enzymes, and we see the results here of each test. So iodine test resulted in a brown/orange color. Now why is it brown/orange? We don't know why is it orange, right? We typically think of the negative result as yellowish brown. Maybe it's because of the other things inside here that mess with the color. So what's clear is that there is no starch. So something must have digested the starch and that's why it's gone. Okay, so the only thing that can do that is amylase. And that's why the student is right to indicate that amylase is present. Now, if you digest the starch, what do you get? You get maltose. Maltose is a reducing sugar, right? So if maltose is in here, when you do Benedict's test for reducing sugars, you will get a positive result, which is why you get red precipitate, reducing sugar is present, which confirms amylase is present. Now, for why it says burette test, and it gives us blue color, which is negative, right? Because positive is violet. But you notice they added protein. So somehow the proteins have been destroyed, right? Proteins gone, broken down. Okay, so what can break down proteins? It's called protease, right? That's why the student said protease is present. Now, there's something I disagree with the question. If protease is present, after it destroys the protein, now what? Now the protease is still there, right? And protease is made of protein because it's an enzyme. So actually, it should still reflect a positive color. Okay, but maybe the setter's intention was that there is very little amount of protease, and that's why it's still not detected, even though there is protein inside. Okay, because yeah, it's true that the test only will detect when there's a significant amount, then it will actually show a color change. So, okay, we'll grant that protease is still present. Then Z, ethanol emulsion test, white emulsion, which shows that fat is present, okay, and therefore lipase is not present because it was not digested. Fat was not digested. So yes, the answer is B. Okay, so looks like this student has done it in our class before. Yeah, it's actually really similar to a past TYS question that we've already gone through in our classes. So we are supposed to compare CO2 concentration. Now, blood from the heart going to the lungs, why? To get refilled with oxygen, right? So this is the highest CO2 in all the alphabets that they gave us. Highest CO2 is present in Z. Okay, it's actually the pulmonary artery bringing the deoxygenated blood to the lungs to get refilled with oxygen, remove the CO2. So we know that here, U is actually the aorta and it will have the lowest CO2. So that's already in my mind as I look through the options. Now, comparing lower CO2, U, so I know one is correct because for sure, U is already a lower CO2. Okay, so it's trying to ask us whether we see this correct comparison. And let's just jump to 4. 4 says that Z has higher. Of course it's correct. Okay, so I already know that 4 is correct. Here, number 3, U is lower. Of course it's correct. So the only thing that I now have to look at is number 2. 2 says that X has higher CO2 than W. Is that true? Yes, because as it passes through any organ, except the lungs, because the lungs removes the CO2, when it passes through any other organ, like kidneys, it does aerobic respiration. The cells need to do this to just stay alive because they need to release energy for their cellular activities. Right? A cell is like a city. How much energy and electricity needs to power a city? A lot, right? So this constantly happens. Even at night when the city is so-called resting, it still needs electricity to survive, to run. So same things happen for all our organs, even when we are at rest. As long as they're living cells, then there will be aerobic respiration. Also, kidneys, selective reabsorption in the nephrons needs active transport, which needs energy. Okay, so that is multiple reasons why they will use aerobic respiration and CO2 produced as a byproduct. That's why all organs will result in more CO2 in the blood after the blood has passed through, except, of course, for the lungs, because the lungs, like I said, it removes CO2. That's its job. So number 2, is that true? That X is higher? Yes. So the answer is 1, 2, 3, and 4. So A is correct. Now, this is very close to a past DYS question. But I warn you guys, don't get complacent because they like to tweak small things. Like this question is not exactly the same. So some students will be like, oh, I remember the answer was 1, 3, and 4, so I'm just going to choose B. Yeah, you have to be careful because they can take inspiration, but they may have tweaked something. Okay, question 10. Which side of the heart is X on in the first place, right? I see that this is really thick. That tells me this is the left ventricle. It needs to have a very thick muscular wall to generate high blood pressure to pump to and around the entire body. Whereas this side has a very thin wall. So I know this is the right ventricle. It doesn't need to generate that much pressure because it's just pumping to the lungs and back, which is a much shorter distance. Okay, so if this is pumping to the lungs, when the blood goes out here to the lungs, this is called a pulmonary artery. Okay, so let's look at the name. Yes, pulmonary artery. Type of blood content is deoxygenated because it's going to the lungs to refill with oxygen. And the direction is to the lungs, so A is correct answer. Okay, question 11 is a harder one because it requires you to have good knowledge about the cardiac cycle graph, and they didn't provide it here. So even though they didn't provide the graph, but they still provided the pressure, where you can see in the left ventricle and the aorta. So suddenly, left ventricle pressure goes up like crazy amount, right? So we know what's happened is that ventricles have squeezed. And now when they squeeze, shortly after, it will force open the aortic semilunar valve, which is the valve at the base of the aorta, right? This is just the aortic semilunar valve, will be forced open. And that is why when this high pressure blood rushes into the aorta, we notice that there's a huge increase in pressure. So 0.2 around here is when semilunar valves open. Okay, so our student here has remembered our memory shortcut, which is a cool scientist owns sick cars and outfits, right? And so he remembers the 4 locations of when valves open and close in the cardiac cycle graph. So I edited his graph a bit. It's not really to scale. Here's when the semilunar opens, and this one is when the semilunar valve closes. So I'll show you from our notes. And our notes are free to the entire public, by the way, even if you're not a TBT student. The link is in the description if you don't yet have our free O-level /G3/Pure Bio notes. So here it is. This is a more accurate graph. And you can see that indeed, this is the time range here to here that the semilunar valves are open. Okay, so the memory shortcut is this. A cool scientist owns sick cars and outfits. And it tells you at all these 4 points. How do we know where the 4 points are? It's whenever there's an intersection. Intersection in a graph pressures. So if there's an intersection, some valve event is happening. Okay, and these are the 4 valve events. That's how to remember it. So let's go back here. Is it true? At that time range, B? Yes. So B is the best answer we can give. Okay, question 12. The diaphragm and intercostal muscles when breathing in. So when you just test yourself, you can do this in an exam and they cannot fault you for cheating because you can use your own body. So you just breathe in and you'll feel that at the bottom area, kind of like in the middle of your body, the diaphragm, you will feel some tension there. Okay, and that's your diaphragm working hard to contract. So when I breathe out, I feel like in this midsection here, the diaphragm, I feel this like tension is now relaxing. So that's actually my diaphragm I'm feeling, that I feel it straining a little bit when I breathe in hard and I feel the tension relax when I breathe out. So the diaphragm is contracting when we breathe in. Okay, now the second one says internal intercostal muscles. Now, intercostal muscles are in between our ribs. Okay, so I'll just draw two ribs here, right? And then the internal ones are on the inside, facing our lungs, and then the external ones are on the outside. So what do the internal and external intercostal muscles do? I know it's a bit confusing, right? Internal, intercostal. So inter means in between, just like intermolecular, intercellular, right? It means in between. So between what? Costal here just refers to the rib bones. So between the ribs. These muscles are found between the ribs, and they're on the inside facing one, facing the lungs. That's why they're called internal intercostal. So the memory hack to know what they're doing is they are named for their jobs. So in the internal one, the job, when it activates, right, will pull the ribs to the inside. And the external one, when it activates, will pull the ribs to the exterior, to the outside. So now you test. You breathe in, your ribs move out, right? So who needs to activate here? The external needs to activate, needs to contract to pull your ribs out. Does the internal want to pull the ribs in now? No, right? We're breathing in, ribs are going out. So these guys will be relaxing. They will not do their job now. They will relax because we don't want to be pulling the ribs in. So that's why answer is relax. And B is correct. Okay, question 13. Pharynx is actually higher up because pharynx and larynx, some students get confused. Pharynx is higher up. It's kind of like the back of our mouth, and we don't see that here. So number 2 is the larynx. 2. Trachea is the tube. Here, number 1. And then we get to the bronchus, right? 3, plural is bronchi. And then bronchioles are the smaller branches from the bronchus. Now, bronchiole, does it sound cute? Sounds cute, right? Like a cute term. So it's a tiny chi. It's kind of like tiny bronchi. So it's a small, tinier branches. And that is how I remember it has to be number 4. Okay, so is that 2, 1, 3, 4? Very good. So the answer is C. All right, aerobic respiration in muscle cells. Now we're thinking of two possibilities, aerobic and anaerobic. Okay, now aerobic respiration, the student has written correctly. It's glucose plus oxygen. It gives us CO2, carbon dioxide, and water. Okay, but anaerobic respiration, what's the equation? It's like this, glucose to lactic acid, okay, plus a small amount of energy. Actually, this one need to add plus large amount of energy. Okay, so now the fact that they gave me 4 elements like this tells me it cannot be the second one. So it's not anaerobic. Okay, this one's out. So if it's aerobic respiration, there'll be oxygen, glucose as the reactants, giving us CO2 and water. Technically, they should add large amount of energy. Okay, so the answer is D. Okay, so this is a very commonly asked question from our own students as well. So we went through this. It's a past TYS question. We went through it in our DBT classes. So I'm going to explain this to you from scratch as well. So remember the kidney's job. What is the kidney's job? It's like a filter. So it filters out all the waste in our blood and returns everything that's clean back to the blood. Now you ask yourself, is the waste going to be a large proportion of the blood or small proportion? Think of it like a fish tank. You know, in the fish tank, it gets dirty over time. So you put a filter that will suck in the dirty water, take away the waste, and it will release back clean water. So you ask yourself, in the fish tank, is most of it the poop of the fish? Or is it just a minority in the water? So the answer is a minority volume. Most of it is just normal water. So I can now draw kind of like a graph. This represents the total volume that is the total blood that goes into the kidney, represented by X. Okay, so X going to the kidney is actually the renal artery, right? Artery is away from the body, so going to the organs. And then Y is the renal vein. Okay, and veins have a larger lumen. Like you can see here, Y is thicker, veins have a larger lumen. Although the walls are thinner, but the lumen is thicker, which also confirms that is the renal vein. So X refers to all the blood, the dirty blood going in. Just like in the fish tank, all the dirty water goes into the filter, right, over time. Then there'll be a small proportion that will be removed. This is the waste. Okay, maybe let's say it's 1 unit here, and this is Z. Okay, and I'm just giving example proportions. In reality, the waste is even less than what I'm giving as an example. Okay, then this whole remaining area is Y, right, which is the cleaned blood that returns to the rest of the body. So I'm going to give Y 9 units. Okay, total X is 10 units, all right? These are just arbitrary numbers which help us with answering these kind of questions a lot better. So now they ask us, which is true about Y? Now we know that Y is 9 units. Okay, given my numbers that I give as example. Is it greater than passing through X? X is 10, right? So obviously not. It's not greater, it's less. Then it's less than Z. Now Z is 1 unit. Is 9 less than 1? No, right? So it's very easy that it's not less. And then it is much less than X. So is 9 much less than 10? No, right? It's slightly less than 10. Okay, then last, which is D, is the answer. 9 is slightly less than 10, right? It's true, slightly less, but greater, much greater than 1 unit. Yes. So now when we put in the numbers, it becomes very easy. When you just slot in the numbers, which option is wrong, it becomes so apparent. So use this hack when doing MCQs. Where does ultrafiltration and selective reabsorption occur? So ultrafiltration is due to high blood pressure. Now, where would experience the high blood pressure is the glomerulus, which is X. Okay? This is where it experiences the high blood pressure, and therefore ultrafiltration happens here. You could say it's in between X and Y. But does it happen at Y? I think no, we cannot say that. So it does not happen at the Bowman's capsule. So Z is now the PCT, proximal convoluted tubule. What does it reabsorb? All glucose, all amino acids, some mineral salts, and some water. Okay, if you have our free notes, we actually put a memory shortcut of how to memorize the what is absorbed at PCT, loop of Henle, DCT, and collecting duct. Okay, so PCT, we remember productive proximal. This person is very productive, they take in a lot of different things. And that's why PCT is actually one that takes in the most amount and different types of substances. So Z, Z is selective reabsorption. Okay, then it just has to be D because we know that reabsorption does not happen at the Bowman's capsule. So D is the answer. Okay, question 17. So it says here that water potential of the blood increases due to drinking. So I'm actually going straight for the correct answer first. Actually, this drinking sounds like drinking alcohol, right? Drinking, no, but I think it's drink water. So yes, if I drink water, water potential of my blood increases because now there's more water in my blood. Then ADH, right? Good. Our student remembers we teach ADH, the memory hack for what it does is like it's water wally. Water wally is always about save water, save water. So now that we have so much water in our blood, what is the body going to say? Save more water? Seriously? Or actually, guys, we can remove the extra water. It's going to say remove, right? So it's going to calm down on the water wally. It's going to be like, hey, water wally, let's not go crazy. So less water wally are released, less ADH. So this is true. And then this will lead to reduced reabsorption of water. What does this mean? Reabsorb from where to where? Actually, you can look up here. Reabsorb from, so there's blood capillaries going all throughout, right? There's a network all around the nephrons. So reabsorb means from the filtrate, go back to the bloodstream. So if it doesn't go back, what will happen? When it passes through all the way, it will go down eventually to the bladder and then it will pass it out. When we reabsorb less, what does that mean? We are letting more water be lost in the urine. So that is true, right? Because we have too much water now. We don't want to take back all the water. We just let it be lost in the urine. So D is correct. Okay, let's look at C. Water potential of blood decrease due to sweating. Yes, this is true because when you sweat, you lose some water and therefore the amount in your blood decreases. So more ADH is released, yes, because the body's like, oh no, not enough water. We need more water, Wally. Please tell the body to save water. That will lead to increased reabsorption of water. Okay, I notice I'm doing from down to up, so let's just continue. B says increased body temperature. Okay, so now you're too hot, right? Blood vessels near the skin surface, to be more accurate, arterioles near the skin surface will dilate. That is true. So the memory hack we teach our students is cold, constrict. If you feel too cold, your arterioles in the skin will constrict. That reduces the blood that can go to the skin, and less warm blood near the skin means less of a chance the heat will go from the skin to the surroundings. So yeah, this is true. It will dilate when you're too hot and cold constrict, right? Then you have increased heat loss. Because you're too hot, you're trying to increase how much heat you lose. So you can come back down, cool back down. Okay, and then A, decrease in body temperature. Okay, so now you're cold. Then cold constrict, very good. Then I would have a decrease in heat loss. Because I'm already so cold, why am I trying to lose more heat? I'm trying to reduce the heat I lose so that I don't freeze. Question 18. So products of digestion are absorbed from the ileum, which is a part of small intestine. Blood from this small intestine now is full of nutrients, right? It enters the liver from hepatic portal vein, which is the green one I drew here, okay, and mixes with blood from the hepatic artery. Hepatic artery is here, right? It gives oxygenated blood to the liver. So now, if you eat a lot of carbohydrates, okay, we can expect a lot of glucose to be in this green hepatic portal vein. Then how will the composition of blood change? This is 2 hours after the meal, so it's quite near to the meal. I expect a lot of glucose will be here. So I'm going to write here high glucose. Okay, this one has high oxygen, low CO2. This one has high CO2, lower oxygen. Okay, good. So 2 hours after the meal, high glucose enters the liver. Now, one of the liver's job, right, is glucose regulation. So the memory shortcut we teach to our students, which is also in our notes, okay, so please get our free notes in the description, is don't have bag, DH bag, don't have bag. Like imagine you left your bag in the MRT, then you're like, oh no, where's my bag? Oh no, the train's gone already. I don't have my bag. So DH bag, don't have bag, okay. D stands for detoxification, and then H is hormone breakdown, B is bile production, A is dealing with excess amino acids called deamination, G is glucose regulation. Okay, so because of the glucose regulation, what will the liver do and how will the blood here in the hepatic vein when it comes out of the liver be like? It will be lower in glucose because the liver sees like, whoa, hold up, we have so much glucose coming in, I can't just let this run around the body, so it converts it to glycogen and stores it, right, reducing the glucose. So it removes glucose. Now insulin, this is strange. Insulin is produced by the pancreas, not the liver. So we shouldn't be adding, liver does not add any insulin, okay? So this is out. But why would it be removed? Because of hormone breakdown. liver constantly breaks down all hormones. Why? Because if you just let the hormone run around the body all the time, then the old message, which is now irrelevant, will constantly be pronounced again and again. Then the cells will be doing the wrong thing, the outdated thing. So the liver constantly destroys hormones so that if there's any new message that needs to be sent, new hormones must be produced, sent around the body. Okay, the liver ensures there's a finite time where the message will be running around the body. It prevents it from constantly running around the body that will cause havoc. Okay, so yeah, it will remove all hormones to some extent. It won't be completely, but it will destroy some hormones always. Now, oxygen. The liver is a huge organ. Aerobic respiration is happening. To keep the cells alive and to do all their jobs, they need to consume oxygen, so they will remove oxygen from the blood. Then urea. Now, high glucose, but also high high percentage of carbohydrates. It doesn't tell us whether there's protein in the meal, but looking at how the direction of the question, right, 3 ticks already, is probably B. Okay, so I'm going to assume there is also some amino acids absorbed, right? Maybe there's some protein in this meal. So the liver's like, wait, hold up, we got excess now. What does it do? Deamination. Okay, when it does deamination, it's going to break the excess amino acids, remove the amino groups and convert it into urea. Urea is now released in the hepatic portal vein, so you have higher urea here. Okay, so urea is added to the hepatic vein. Added. Yes, so the answer is B. Question 19, fantastic. Our student used our memory shortcut here. So how do you know what the radial and circular muscles will do of the eye? Well, these are muscles of the iris. What will they do when you're going into a dark room? So the memory hack is that the radial muscles are like a little kid. They're scared of the dark. So because they're scared of the dark, if you turn off the light, they'll be like, oh, what's happening? Then they'll tense up, they'll contract. You know, they're scared. So they contract. Okay, and then circular, they just do the opposite of radial, so they relax. Okay, so radial contracts, circular relax, answer C. Okay, question 20. What happens to these 3 hormones after a long period of vigorous exercise? Now, if you are exercising really vigorously, you're going to sweat a lot. Right? You have experienced this, maybe 2.4 run, right? PE lesson. You're gonna sweat so much, so you're gonna lose water. So if you lose water, the body's like, okay guys, we need to save water, save water. So they will want more of ADH. ADH is the water wally mascot, very good, our student remembers. Okay, so ADH drop is to tell the body, hey guys, let's save water. So do you want to save water? Yes, right? You've lost a lot of water, you want to save. So ADH will increase. Okay, next, glucagon and insulin, we have to look at the blood glucose levels. So this is the blood glucose, this is time. Now, when you exercise vigorously for long periods, you're going to see a drop in the glucose levels more and more and more. Okay, but the body doesn't allow that, right? When there's a drop, glucagon comes out when your glucose is gone. So that's how I remember what glucagon does. Glucose gone, glucagon comes out to solve the problem. So it will actually bump it up back to normal, okay? So when this happens, when you have less glucose. Now, what's insulin's job? Insulin's job is to get the glucose to go into the cells. Because like, oh, there's too much glucose in the blood. Let's push it into the cells, the liver and muscle cells. So we have the opposite problem here. It's not that we have too much glucose. We have too low glucose. So do you want to push more glucose into the cells? Then you have an even bigger problem, right? So we want less of this, pushing glucose into the cells. We want less. So I'm going to see a decrease. Okay, so overall, the answer is increase, decrease, and the answer is B. Good job. Hey guys, today's sponsor of the video is me. As of making this video, that BioTutor is Singapore's only O-level pure bio specialist. You mean you don't teach any other subjects or syllabus? Yup. That's how we've been able to make so much resources just for your syllabus. Example, concise notes without all the extra fluff. Commonly asked questions that come out under every topic, so you know the exact keywords and phrases to score full marks. Didn't pay attention in class? Crash course clips to the rescue. We've recorded explanations for every concept under every chapter. Find bio a chore to memorize, flashcards on your phone, which makes it feel like a game. So if you need help with O-level pure bio, what are you waiting for? Click the link below this video or go to thatbiotutor.com. Now back to the video. Question 21. So the student is looking at the far object and then chooses to focus on a near object. Alright, the memory hack that we teach our students is, when looking near, what do your parents say? Stop looking at your phone, right? Stop looking so near. You're straining your eyes. You need to give your eyes a chance to relax. Look far and you can relax your eyes. That's what they always say. And actually there's a truth to it, because the ciliary muscles will contract when you're looking near. But then when you look far, they relax. So it's kind of true that you're relaxing your eyes. So when you look near, ciliary contract. Now what happens to suspensory ligaments? Okay, look what happens when ciliary contracts. It moves closer towards the lens. So this will naturally cause the ligaments, which are kind of like ropes, to then become slackened. Okay, so become loose, yes. And then the thickness of the lens, because the lens is flexible. So when the ropes are pulling it, the suspensory ligaments, right, which they're not actually called ropes, they are like ropes, when they pull on this flexible lens, it stretches it out thinner and it'll be less convex. But now when you release the ropes, the lens goes back to its original thicker shape. So it becomes thicker and more convex. So thick, and therefore the answer is A. Question 22, which is true about all antibiotics. Now what exactly are antibiotics? Think of them as kind of like an insecticide, but for bacteria. So it is a poison chemical that attacks bacteria only, right? It's a poison. So it's not the same as antibodies. Antibodies are proteins. Antibody, okay, are proteins made by immune cells, more specifically the lymphocytes. They are made of protein and they will bind to antigens on any type of pathogen. It can be bacteria, virus, doesn't matter. So not to be confused with antibodies. Antibiotics are just one chemical. Antibody is a whole protein. Okay, so does it stop virus? No, antibiotics don't attack viruses. It is useless on them. B, destroy the protein coat. Okay, protein coat is actually the virus component, right? It's the outermost, like the shell of a virus. So bacteria don't have protein coat. They have cell wall and they have a cell membrane in their surrounding. So B is also wrong. And then antibiotics kill bacterial cells, yes, without harming any cells in the body. Now this is kind of vague, but I think it's talking about human cells. And yes, it does not directly affect any human cells, because just like insecticides are specific to the insect, if we get a low concentration of insecticide in us, we're okay because it targets insect cells, not human cells. So same thing, it targets bacterial cells and not human cells. That's why you'll be fine. So C is correct. And then treat any disease, not only bacterial diseases. Question 23. This is about the amount of chloroplast concentration in each of the leaf cell types. So plate mesophs are the ones which are vertically arranged like that, and they have the most number of chloroplasts because their main job is photosynthesis. So B is the answer. Spongy mesophs are irregularly shaped, and they don't have as much chloroplasts. Their job is actually mainly gas exchange, because they are the ones that make up the intercellular S spaces for gas exchange. Then upper epidermis, epidermal cells have 0 chloroplasts in them. 0. Okay, so this is 0. Then lower epidermis also has 0, but guard cells have chloroplasts because they need chloroplasts to photosynthesize in order to respond to light. Okay, so how does it work is they respond to light. When light shines, they photosynthesize, releasing energy. And they use this energy to then make themselves become turgid. Okay, so I'm skipping past a lot of steps in between because it's our syllabus. But basically, they respond to light to become turgid. So they must have chloroplasts because that's the only thing that can do some kind of chemical reaction when there's light. So low epidermis has very few. This one has the most. Spongy has some. And this one has 0. Yeah, epidermal cells. Because upper epidermis, we're assuming no guard cells. Now, in reality, there are a few stomata on the upper side. Most stomata on the lower side. So the answer, yes, it is B. Question 24, which is a leaf adaptation to increase gas exchange during respiration. Okay, so it's trying to take in oxygen from the air outside, right? You might be thinking, wait, how come leaves respire? Yes, are all plant cells. Since they are cells, they need to survive, they need energy to survive for their cellular activities, and that requires aerobic respiration. So most plant cells have mitochondria and they do respiration. So now, the presence of waxy cuticle on upper epidermis, this is actually to reduce water loss. So it's not about respiration, it's not related. Then B says, the structure of the vascular bundle. Now the vascular bundle, what it does is it transports, xylem will transport water mineral salts, and phloem will transport manufactured food substances such as sucrose amino acids. So that's actually about transporting substances. It doesn't help with respiration. The taking in of oxygen is not relevant. Okay, then C says the structure of the spongy mesophile, because like I drew some spongy mesophile cells here above, and because of its irregular shape and this arrangement, right, creating many different air pockets, intercellular air spaces, if you were to measure what is the surface area to volume ratio, okay, the surface area is very high because of all these irregular shapes. And there's these like nooks and crannies everywhere in the intercellular air spaces that increases the surface area for taking in oxygen. Okay, same thing for taking in carbon dioxide. It is also true. So yes, this structure actually increases the SA to V, which increases the rate of oxygen diffusion, diffusing in. So first it has to dissolve in a thin film of moisture, then it diffuses into the cell. So C is correct. And then there is no waxy cuticle. This is actually false. There is a waxy cuticle on both the upper and the lower epidermis because we don't want to lose excess water unless we are losing water through the stomata. Now that is intended by the plant. Question 25. Carry out photosynthesis, you produce a carbohydrate called glucose, right? That is the end goal of photosynthesis. Now what do we use this for? Aerobic respiration. Yes, so we release energy. Now glucose can be converted to a lot of other kind of molecules as well through enzyme reactions because it's made of C6H12O6. Okay, so to make lipids, what are lipids made of? Well, lipids actually is a big category, including fat molecules, right? So when you think of fats, we know that fats are made of C, H, and very little O. So do you notice that it's the same elements, C, H, and O? So the enzymes, there are many enzyme reactions and many enzymes involved in breaking apart the chemical bonds, rearranging, and then eventually forming fatty acid chains, as well as glycerol, right? So a lot of oxygen atoms will be removed. That's why very little O. So this is true, yes. We put a tick. And then now stored as starch. This one is actually quite easy because glucose, many glucose units, there will be an enzyme that will put them together as well. Then you form starch because starch is just made of many glucose units. Okay, then the first one, convert to protein. So glucose to protein, well, proteins are made of amino acids. Amino acids, what's the biomolecules? C, H, O. All these are common, right? You notice? Then N, nitrogen is a new one, and sometimes sulfur, S. So where does the N, the nitrogen, come from? It comes from nitrate ions, mineral salts basically, such as H3 aqueous. This is in the soil. And there's other forms like NO3 minus, these kind of things. NH3 aqueous. I don't know if there's going to be ammonia directly in the soil. Okay, so I'm going to remove this. Sorry, I'm not chemistry expert here, but I'm pretty sure this one is legit. So nitrate ions in the soil, that is how the plant will take that in, then bring up through the xylem from roots to leaves. At the leaves, enzymes will deal with this nitrate ion, take out the nitrogen, put it into the amino acids, and that is how we can turn glucose into proteins. Because after all, plants don't eat protein, they don't digest, they don't absorb amino acids like we do. So for us, we don't have to make amino acids because we just absorb it directly from our digested food. But plants, the only thing they can make is glucose. So they have to take this glucose and then turn it into all other kinds of biomolecules. So they are able to do this. And so it's tick, 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 everything is correct. So the answer is A. Yes. Question 26. Companion cells are found in phloem. They are the companions of the sieve tube elements. Why do sieve tube elements need companions? Because the sieve tubes, they have lost most of their cell parts. And their job is they've become very specialized to just being a tube, allowing the food substances to just go through. And they've lost all their mitochondria as well. It's a sacrifice in order to make more space. And that's why the companion cells which are beside have adaptation to be very high in the number of mitochondria, okay, to do aerobic respiration, not just for itself, but also to give the sieve tube element. And translocation, which is the movement of things through the phloem, requires energy, okay, requires active transport. So that is why companion cells, it is correct, they control translocation, okay. So how about ion uptake? Nope, this happens at the roots. Photosynthesis, nope, companion cells are hidden within the vascular bundles. They're not exposed to light, so they have no job in photosynthesis. They have no chloroplasts. Then transpiration, that would be the loss of water, right? And now water would be going up the xylem, not the phloem. So companion cells, again, not related. So the answer is C. Question 27. So let's figure this out together. The contents of plant cells have negative 400 kPa water potential. Now, some of you might have been confused. What is this negative number? Why is it even negative? And so, good, our student here has remembered that we've mentioned this before in class, that how the scientists measured water potential. I don't know why they did it this way, but they decided that pure water, okay, this is 100% water, they tagged this to 0 water potential, 0, okay? So anything which is less than 100% water, example, if I make coffee with 90% water, okay, 90%, and 10% coffee powder, 10% coffee. So now obviously this has less water potential than the pure water, right? Therefore it will be negative example 200, okay, kPa. This is just an example. I don't actually know if it's the correct unit exactly. But the moral of the story is that everything is always negative. Water potential is always negative. The highest you can get is 0, which is pure water. So now the plant's cell has a negative. This makes sense because plant's large central vacuole contains some dissolved mineral salts, dissolved sucrose, amino acids, manufactured food basically. So it will not be 100% water, it will be less, and therefore water potential will be negative number, just like my coffee here is a negative number. So they place it in a solution and it loses even more water. Okay, so I can draw, this is the petri dish, this is the cell, the plant cell, okay? And water came out. So which one has higher water potential? This one is higher, this one is lower water potential. So the solution must have less, right, lower than the plant cell, lower. So in the number line, if you think about it, right, what's even less than negative 400? The only option is A. So yes, A is the correct answer. Question 28. What will cause a plant to lose most water? Humidity has to be low. Why low humidity? So that steep concentration gradient of water vapor between the intercellular spaces, which will be very high concentration, and the outside, low concentration. Now, humidity just means concentration of water vapor in the air. So if inside the leaf, right, let's say this is exaggerated intercellular space, very big, has a lot, right, high concentration, because the thin film of moisture that surrounds the lines, the intercellular spaces, is constantly evaporating into water vapor. Then now outside, if it's low humidity, meaning low concentration of water vapor, low humidity, now there's a big gradient. There's a very steep gradient. So this will come out really fast, right? Diffusion will be very fast. Okay, and if you're losing water vapor very fast, transpiration will be higher, will be increased. So we're looking for low humidity. Right, next one, temperature. Now, again, when it is evaporating, temperature will cause the evaporation to be faster, right? You just think about a puddle outside on the road. If it's very cloudy, right, dark, the puddle will take super long to evaporate. But if it's like, whoa, really noon sun, very hot, in a few hours, the puddle is completely dried. So we intuitively know that the higher the temperature, the faster evaporation. So if this is evaporating faster into water vapor, then it will lose water faster. So we want high. Now, wind speed is just the opposite of humidity. So the higher the wind speed, then the lower the concentration. Okay, so we want high. Because if the wind is high, it will blow away the water vapor surrounding the leaf. So we want high temperature, high wind speed, and low humidity. C is the answer. Okay, question 29 is a really fast one. Biodiversity. What does that mean? It's the range of species in an ecosystem. That's the definition. So if I have more types, that is greater biodiversity. It's not about how many, it's about how many types. So yes, the student has rightly pointed out that B has the most variation in the species, most number of different species. So B is the correct answer. Okay, question 30. So which arrows involve release of energy? First one, carbon dioxide in the air becoming carbon compounds in plants. This is CO2, gaseous. This is glucose, right? And all the other carbon compounds that glucose can be converted into. So yes, this is photosynthesis. That is the correct process. Is that release of energy? Actually, it's the opposite. Light energy is trapped, right? It is converted to chemical energy. So it's actually trapped, not released. So one is wrong. And if one is wrong, then these are all wrong. So the answer has to be D. Okay, so this is a very nice question because it's not so ambiguous. Then for number 2, statement 2, honestly, this one is more ambiguous. Because you could say that when animals eat the plants, right? And maybe now it is stored in starch. Animals will break down this starch into glucose and use it for aerobic respiration. So that is a release of energy. So that's why this one's like, is it a yes? I'm not sure. But then again, to carbon compounds in animals, now it becomes glycogen. So if they just broke it into glucose, then put this glucose into a long chain glycogen, there wasn't a release of energy. It's just converting from one form to another. So that's why 2 is a question mark. I don't know if they actually release the energy in the starch. Okay, so I was actually leaning to yes, they would release the energy as starch from starch, but clearly there's no 2, 3, and 4. There's only 3 and 4, right? And by elimination, I already know answer is D. So now let's look at 3 and 4. 3 is fossil fuels. Yes, you combust. This releases a lot of energy, right? Heat energy is the main form. And then carbon compounds in plants, aerobic respiration gives you CO2, correct? That also releases energy. Okay, so that's why your answer is 3 and 4. Okay, so we got bacteria who are genetically engineered to produce for us human insulin as part of their metabolic process. How can we use them? So A says, eat and digest the food containing transgenic bacteria. So if you eat the bacteria, do you get the benefit of supplementing your bloodstream with insulin? Because diabetics have a problem with they cannot produce enough insulin into their bloodstream. So if we eat the bacteria, maybe there's insulin molecules in the bacteria because it's producing insulin as part of its processes, right? But when we eat the insulin, it's a protein. Okay, so any protein that goes into our stomach, what do you think will happen to the protein? Will it stay intact? It will be destroyed by protease, the enzyme. It will also be denatured. Now, you only learn in your syllabus denatured enzymes, right? But actually, proteins also can get denatured and messed up by the low pH. So we also do not directly absorb proteins straight into our bloodstream. First, we digest them into amino acids, the small molecule. Then we absorb the amino acids. So that's why this is not a good way to get insulin. You cannot just eat insulin to get it into your bloodstream. You need to directly inject it, which is option B, which is the correct answer. Okay. So next one, inject the bacteria into the blood of the patient. That will cause complications and the immune system will be like, hold up, who is this guy, right? And then they will launch an attack to kill the bacteria. So this is not sustainable. Soon the bacteria will be gone. And then last one, replace defective cells in the pancreas with the transgenic bacteria. We have the same problem, right? So these two, the immune system will launch an attack, right? They will launch an immune response. All right, so let's unpack question 32. I'm going to draw 2 chromosomes. These have the same shape, length, size, and same genes at corresponding locations. Let's say both of these are called chromosome number 1. So in our normal diploid cell, diploid means having 2 sets of chromosomes. Most of our cells have 2 sets of chromosomes. One set came from the father, one set came from the mother. So that's why we have 2 of every chromosome. That's why there are 2 chromosome 1s in most of your body cells. These are called homologous chromosomes. Okay, so that's actually option D, talking about homologous chromosomes. And now, if I were to untangle each of these chromosomes, what would I get? I'll get an extremely long DNA. Extremely, it's super long. It's one super long strand of DNA. So what are chromosomes? They are made of DNA, right? It's an extremely long strand. Now in this DNA, there are several sections that are meaningful. Just like in a super long bio textbook, there are many sentences that make up the textbook. So here's sentence 1, sentence 2, sentence 3, and so on. Actually, chromosomes have tens of thousands of these meaningful sequences called genes. Okay, so I'm going to now highlight some of those genes. So here's gene A, and on the other side, at the same position, you will have that same gene. That's why I'm using red color as well. Okay, so I'll just show a few more. The blue one here, let's call it B, will also exist on the other side. And then C at the same position, D at the same position. Okay, so Now let's look at option A. A chromosome contains a DNA molecule for each gene. That's kind of true. You see, for each gene here, right, there is one DNA segment, but not a DNA molecule, because this whole thing is one molecule, one very long molecule. So that's why it's a bit weird. If it said it contains a sequence of DNA nucleotides for each gene, I would say that's quite accurate. Okay, but the phrasing right here is not accurate, so wrong. Gene is a section of DNA. Yes, I agree with this. It's a section. It's a sequence of nucleotides found at a specific site on the chromosome. Yes, this is the specific site for that gene, which we are calling gene A, responsible for trait A. And it's found at the gene locus. This is one gene locus. That just means the location, gene location on the chromosome, gene locus. Okay, so at a specific site, gene locus on the chromosome, so B is the answer. Then each strand of DNA molecule codes for different polypeptides, because each gene codes for a specific polypeptide. So when you see the word codes for, the phrase codes for, it's trying to say that this after the word codes for is the product. What is the product it's coding for, right? So each strand of DNA molecule, actually each gene codes for the product. The product is a polypeptide. Okay, so a gene is like a recipe, and then the polypeptide is the food, the dish. So let's say it's a recipe for burgers. If you follow it, it will code for a burger. It will instruct a burger to be made. But obviously, our genes don't code for burgers. They code for proteins. So now D, homologous chromosomes always contains identical alleles. So you can see here, what are alleles? Alleles are alternative forms of the same gene. That means at this position, right, at this gene, I can have an alternate version that is similar but not identical. Why are they not identical? Because in the other strand here, in this other super long strand of DNA, if I look at the same location, let's say here, right, maybe the sequence is AATC, this one is ATTC. So there are these small changes, right? Of course, the sequence is many thousands long. Each gene is usually thousands of bases long. I'm just showing an example, super short one. So whenever there are these tiny nucleotide changes, this makes, I call this version 2, I call this version 1, right? There's a slight change now, it's a different version. But it is the same gene because it is responsible for the same trait. What does that mean? Okay, I'll give you an example. Let's say the teacher chooses 2 students to be the bio rep for your class. So both students have the same job, being the bio rep, right? Y'all, 2 of you are supposed to handle all the, bio notes, printing, worksheets. You guys handle. It's your job. Okay, so both of them have the same role, bio rep, and they're supposed to carry out the responsibilities. Now, maybe the first student is very hardworking, right, and does all the job. The second one is a slacker, slacks off, not responsible. Now, if I'm an outsider and I come in and I say, who is responsible for this trait, bio rep? Who is responsible for the job? Both of these people, student 1 and 2, will say, I am. I am responsible for this job. This is my job, my role. Now, if I dig more closely, I'll see that how they perform the job is not the same, right? One is actually doing it, one is not doing the job. But they both have the title bio rep. So here, now we use A, okay? The first one at the top. So on the left, this could be a useful functioning allele. So both alleles, the big A and the small a, on this gene locus have the same job. Maybe the job is, can you roll your tongue? So the first allele is doing its job. It allows the person to roll their tongue. The second one, maybe it's just slacking off. It's not functional and it doesn't help the person roll their tongue, even though that's their job. So that is what alleles mean. They are alternative forms of the same gene. They have the same job assigned, the same trait assigned, but whether they're actually functioning at their job, that is up to the allele. We have to look into the allele. Is it working or not? Okay, so that's why it is not true. You don't always have to contain identical alleles. Question 33, we notice that the parent plant is attached directly to the offspring, and it seems that these are then growing into new offspring plants. And eventually this will break off, and that is how it separates from the parent. So this is asexual reproduction. There is no fertilization. That's how we know it's asexual. And therefore, it uses only mitosis, okay? Whereas sexual uses both mitosis and meiosis. So genetically identical, yeah, because mitosis does that. Produced by meiosis, no. Contain different alleles to that of the parent plant, no. Because since it's a clone of the parent, so all the alleles are exactly the same, then half the chromosome number, no, this is for males, right? And meiosis doesn't happen. So the answer is A. Okay, question 34. Which is true about meiosis in humans? The chromosome number is maintained. Nope. Yeah, the student is right to say that chromosomes number will be half. In fact, in human, 46 is the parent cell, which is diploid. Then the gametes, which is after meiosis is done, is 23 chromosomes. So it is halved. Okay, then statement 2. It involves reduction division. Yes, this is meiosis 1. Meiosis 1 is called reduction division because that is when the chromosome number officially drops by half. It's reduced. That's why you call it reduction, because reduce the number of chromosomes. Then 3. It produces cells that show genetic variation. Yes, the gametes are genetically different. Okay, this is true. They are genetically different. So why is it different? Imagine this. You have a deck of cards. Okay, this is the deck. I'm going to try and draw 3D deck of cards. You have a deck of cards. 46 cards. Now what meiosis does is it randomly chooses half and splits the deck anyhow, right? Just as long as you split half-half, but it can be in any way, shuffle them anyway. So this is a simplified explanation. I know compared to the chromosomes what's going on, but bear with me. So we remove half of them into this smaller pile. Okay, now I have 23 in this one, and the other part 23 as well. And since I can split this deck any way I want, okay, I'm going to use a different color. Yeah, so since I can split this deck any way I want, I can generate lots and lots of different combinations of what cards are in. You know, if this is like a poker playing card set, I can generate lots of different combinations, right? Many possible combinations. That's why there's genetic variation in the gametes. Okay, so 2 and 3 are correct. Answer is D. 35. Which process involves mitosis? Forming gametes is meiosis. Growth of a cell? No, because mitosis means cell division. The cell has to divide and make a new cell. So when the cell just grows from smaller to a bit bigger, right? There's no mitosis. Then growth of an organism, let's say when you were a baby and then now you are a bigger, teenager. So what changed? Is it your cells grew bigger? No, you had to increase your number of cells actually. So to increase the number of cells, you need cell division by mitosis. So C is the answer. Then D says production of haploid cells. Again, that is the same. It is meiosis, same as A. Alright, this question, yes, we've done it before in our classes at TBT because we've explained and went through this TYS MCQ. So this is why, again, I'm reminding you guys, especially those of you who are sick right now, please do all the TYS throughout your year. Like, you can save it for near the end of the year because when it's near O-level, then you practice TYS, but you have to do all the 10 years. Okay, and in TBT, we have past year papers. We have the soft copy of TYS beyond the past 10 years. We have a few extra years and the answer key as well. So yeah, do it and you get free marks when this kind of thing just repeats again. So describe the variation in these birds. Okay, pattern of spots. We look at the spots. Does it fit into continuous or discontinuous? Now, what is the difference? Continuous means a wide range of phenotypes with many intermediates and no distinct categories. Okay, no distinct categories. Then discontinuous variation, you can see few and distinct categories. So here, can I categorize the pattern of the spots? Like they're actually very, they can be here, they can be there. So I'll put this as continuous. Okay, continuous variation. Presence or absence of the ring. Okay, where is the ring? If you see, the males have a white ring around the black eye. Okay, so that is actually the ring that this question is talking about. You notice that it's either you have a ring or you. It's yes or no. It's two categories. That's why discontinuous. Now size. Size, weight, height, these things you already know is continuous, right? Is it all people are either big or small size? Is there big, medium, small size? No, there's a wide range, right, of the size of a person, and same goes for the birds. And it's actually affected by how much they eat, so it's affected by the environment, as well as their genetics. So it's not only genetics or environment, it's both. So this one is continuous. So it should be C, D, C. A is the correct answer. Okay, which term is defined correctly? Diploid means having two full sets of chromosomes, one from each parent. That's the definition. So A is wrong. B says heterozygous. Okay, heterozygous is, let's see, having genes at the same position on each pair of chromosomes. Well, no, heterozygous is when the alleles are the same position, like this one. Let's say it's big B, this one is small b. But heterozygous is about the alleles being different. So if you have big B, that is homozygous. Small b is also homozygous. Big and small, or small and big, that is called heterozygous, because hetero actually means different. Okay, so no, this is not the definition. Then C says phenotype, the observable, the outward traits that you can see as a result of the genotype of an organism. Yeah, C is the answer. And then recessive allele expressed when only one of them is present. Actually, when only both two of them are present, that is when a recessive is expressed. And in fact, this is a dominant allele answer, right? If we don't change this, this is actually a dominant alleles description. Okay, in a family, one parent has blood group A and the other has blood group B. Now the children are A or O. Whoa, interesting. So how do you get blood group O? So for A and B, we don't know exactly the genotype, but for O we know it has to be IO because the O allele is recessive. So now let me just do an example family tree, father, mother, and just two children, okay, for example. So maybe one child's A, one is O, then let's say one parent's A, one parent's B. It doesn't matter whether it's mother, father here. And then I know this is IO. And that tells me each parent must have given one IO allele. So I'm starting to work backwards. And I see that, okay, that means how would the person be A? Because they have a dominant A allele. And then how would this person be B? Because they have a dominant B allele. Okay, now I'm starting to work out the parents. And then now this child is A, right? So obviously you must have at least one IA, but the mother, in this case, the only thing she can give to make the child A is the O allele, which basically doesn't do anything, right? Because it's recessive. So I figured out the parents, and okay, I don't recommend doing this kind of thing because if the question is a family tree question, drawing a family tree really helps us. So we can reduce our careless mistakes. The parents are going to be A, O, B, O, and then children A, O, O, O. Yes, B is correct. Question 39. Which is the force for natural selection? In other words, it is asking which is the selection pressure, okay? To push for selection towards a certain direction. And actually all of these, right? even if you didn't understand the phrase up here, if you read this, all of these are examples of possible selection pressures. But in this scenario, we know that they evolve resistance to the use of penicillin. So using penicillin is what is the selection pressure. Now, I give you a tip for knowing what this means, right? What does selection pressure mean? It is whatever is killing off some individuals. Anything that kills off some individuals is the selection pressure. So in this scenario, what is the thing killing off some of the bacteria population? It is the use of the antibiotic, right? So that's why C is the answer. So UV radiation is constantly happening, and yes, it can lead to mutation. So this would actually result in random mutations, which lead to variation. Because bacteria cannot have variation, they're not sexual, they're asexual, and that's why the only variation they can get is due to random mutations. This variation can then lead to new traits. Then these new traits are tested against the selection pressure, are tested to see if it's beneficial. So if the new trait can resist penicillin, then it will be selected as, yeah, this is good. This guy will survive and reproduce. This lucky mutation is beneficial and it will stay. So it doesn't answer the question because this is not the selection pressure. UV radiation is a mutagen which increases the mutation rate so that later natural selection can act on it to choose, is this a good new trait that came up? Is this a good mutation? Okay, so B is out. Then D is a use of disinfectants. No. If let's say the question changed it to resistance to alcohol disinfectant, right? If the question changed it to that, then it would be correct. Then you would say yes, use of disinfectants. Okay, but no, the question specified that it's evolving resistance to penicillin. So penicillin is the thing killing off the individuals. And now last question, question 40. So I don't actually like the graph because it's inaccurate. The estrogen peak should be higher than the progesterone, okay? And let me show you, this is from our notes again, our entire content notes with all the memory shortcuts. All these good diagrams are free, right? Just go to thatbiotutor.com/notes, or you can click the link in the description. If you're G3 Bio, Pure Bio, Sec 3, you're turning Sec 4 next year, right? Or you're turning Sec 3 next year, this would be super useful to you. Estrogen peaks higher. This is why I say I don't like this diagram. And it happens around day 13, slightly before ovulation happens. So now I look here and look at the peak. That tells me this is day 13. So R should be ovulation day 14. Okay, and the question asks, when does ovulation occur? The best answer I can give is between R and S. Okay, so B is correct. And this, just sidetrack, some graphs, their questions, right, they will not show you a second peak. They will just show this. Okay, so they won't show a second peak in estrogen. However, it is more factually accurate that there is a smaller estrogen peak at day 21, along with the progesterone peak. So this is actually true. This part that it showed, it is true, but some graphs, you might get no second peak. So don't be afraid if you see that. Okay, so we've come to the end. That is question 40 already. The student got 38 out of 40 for this paper. Very good. We always want to strive for as close to 40 out of 40 as possible because MCQ is only 1 hour and you get 30% weightage, which is incredible, right? Just for shading some OES, you get 30% of your weightage for your whole grade. Yeah, so if you are Sec 4 and you have just finished this paper, right? You've just taken O-levels and finished. Congrats on your freedom, by the way. Enjoy it. And comment below in the YouTube comments, what was your score, right, out of 40? I'll convert this to PDF and I'll upload this on Google Drive. I'll let you guys download it and compare to your own answers. And I'll also be releasing a summary table of all the answers in one place so you can easily mark, okay? For that, see our Telegram channel. The link for how to get this PDF and the Telegram channel link will be in the YouTube description as well. Then, now, if you're Sec 3, then please learn from the mistakes up here. Please learn from how to answer all these questions because they really can repeat very easily. And to obtain our good TYS answers, right, that's actually reliable, the options are actually consistent and make sense, and as well as the open-ended are actually good, unlike the publisher TYS answer, which a lot of students say is not good. You can get that also in our Telegram channel. Just click on the pinned message in our Telegram channel and you'll be able to get that for free.